Okay, my name is Bryce Williams. Welcome to my presentation. Don't mind the background. Okay, so this is my poster. Uh, so today I will be talking about meiosis. So meiosis is basically, put into simple words, it's the splitting of cells. Some people confuse this with mitosis, which is probably also common to hear about, talking about cells. It is not mitosis. Mitosis creates identical cells for the body to grow or to regenerate when you get injured. So it is not mitosis. Meiosis has a genetic purpose. So it happens with, in the case of the human body, it happens with the male sperm and the female egg. So, and it only happens in those cases. So in order for us to understand meiosis, we first need to learn about daughter cells. So what are daughter cells? Daughter cells are these. You see these two little cells. This used to be one cell, but it split, so there are now two. So these are called daughter cells because they're daughters of what used to be one cell. And each of them have these strands of chromosomes. So originally it had 46. As some people may know, the normal cell has 46 chromosomes, but when it splits, it's basically halved, right? So 23 chromosomes in each cell, each daughter cell, and it also has one member of this homologous pair. And so now that we've learned about daughter cells, to start this meiosis process, we need to create we need to have a regular cell again. So a daughter cell goes through this interphase before it gets into meiosis, which basically doubles everything. So the chromosomes are doubled, and it enters into the meiosis process. So meiosis has two big phases. So we're entering into the first one, and the first thing that happens is the prophase 1. This is where everything's doubled, 46 chromosomes, and it says 4N here. This is just, so basically it has two X's. So an X is a diploid, which is 2N, and if it's just a strand like this one, or single strand like this one, it's going to be a haploid, and it's just 1N. So it's 4N in this case, in the prophase 1. This is where chromosomes pair up and start swapping genetic information to create this unique cell. And once it's and it also creates this thing called a spindle. So it kind of what it does is it can contract the cell or like expand it. So all that is going on in the first phase, and then it moves into metaphase 1. So this is when the chromosomes line up in the equator, so it kind of gets into order. And once that's done, the spindle pulls the cell. I think it expands, and we enter into anaphase 1. This is where the cell actually starts to wanting to split. So and then it moves on to telophase 1. So this is where the cell actually is about to split. As you can see, there's this pinch in the middle. It just wants to just go. So that happens, and the cell actually splits into two, entering into the meiosis phase 2, meiosis 2. So once it does that, the whole process starts again. So we enter into prophase 2. Now we got these X's, so 2N, 2N, 1X in each cell. Except what's different from the first one is there aren't going to be any pair ups with the chromosomes because it just only has one X. So then we move on to metaphase 2. Metaphase 2. Same thing. The chromosomes line up in the equator of the cell. Then we move on to anaphase 2. Same thing. The cell wants to split, so it kind of like expands, and then we go into telophase 2. The pinch in the middle, it's about to split, and then finally we get the whole thing both splits, and we get 4 cells. Then the process is complete. We got one N in each, one strand of I guess you could call it haploid. In each cell, each have unique genetics. So the process is complete. We have 4 genetically unique daughter cells.